Hi everybody, and we're back, and I'm super excited today to have the winners of this year's Student Poster Awards, both from SLAS 2025 in San Diego from February and SLAS Europe in my hometown of Hamburg just a month ago. We were very fortunate. We had 6 winners in all, but as anybody who's tried to schedule a meeting with more than 4 people at once knows, it's sometimes a little bit difficult to find the perfect time for everybody, especially as we head into the summer and it's sweltering heat here in Hamburg, of 36 degrees today. So we're going to ask the questions of those who could make it, and let's get started. So first of all, let's take some turns to introduce ourselves. So maybe Toan, we can start with you. Please just briefly introduce yourself with your name, where you're studying, and the title of your poster, just to give us an idea of the breadth of the work covered by the winners today.
So hello everyone, it's very happy to see the familiar face that I saw in February this year again. So my name is Ton Pham. I'm originally from Vietnam and trained as a dentist. Right now, I'm pursuing my PhD in oral biology at Chulalongkorn University in Thailand. So the poster that I presented in SLAS San Diego is about the cellular extracellular matrix hydrogels. It enables oral mucosa on a chip. So alongside my clinical background, I'm working on a research that bridges basic science and translational application. I'm very grateful for the opportunity to connect with the SLAS community and share my experience. Thank you.
Thanks a lot, Toan. And my colleague from the Fraunhofer here in Germany, Silvia.
Hi, my name is Silvia Materna-Reichelt, and I'm doing my PhD with Fraunhofer ITMP. And I presented my poster here in Hamburg. And the title of my poster is Side-by-Side Comparison of Three-Dimensional Drug Screening Assays, Identifies an approach that reduces cost and complexity and increases reproducibility. And the topic is about 3D drug screenings on patient-derived models. And what we pursued was to make it more simple in terms of the model propagation for the drug screen. And then also everyone who works with animal-derived hydrogels knows it's not so easy to handle it, especially in automation setups. And so I introduced synthetic hydrogel, and yeah, this helped really to improve to have the assay at room temperature running. And yeah, that's my
I know it was a hard project, but we'll find out more about that in a moment. So, Lan, would you introduce yourself and your project?
Of course. So hello everyone. My name is Lan Mi, and I am currently doing my PhD at UMass Amherst, and I'm studying in the Department of Chemistry. So my poster title was Genetically Encoded Fluorogenic RNA-Based Bioluminescence Resonance Energy Transfer Sensors for Cellular Imaging and Target Detection. So, which is really clear from this title that we constructed an RNA-based bioluminescence sensor for the detection of some small molecules that's related to some certain kind of diseases, like both in vitro and inside cells. And we're planning that maybe in the future that this kind of method can be used for some high throughput screening for some small molecules for efflux pumps or something like that. Thank you.
Thank you, Lan. And last but certainly not least, Maryam, could you introduce yourself and your work?
Hello, everyone. My name is Maryam Salarian. I'm originally from Iran, and I moved to Germany 4 years ago to start my PhD in biology at Karlsruhe Institute of Technology, which I recently completed. The title of the poster is Miniaturizing Drug Sensitivity and Resistance Testing for Non-Small Cell Lung Carcinoma on Droplet Microarray, which was conducted in collaboration with Thorax Clinic from University Hospital of Heidelberg. So my research focused on developing a miniaturized platform to test how tumor cells from individual cancer patients respond to different anti-cancer drugs. And the goal was to identify the most effective treatments using a very small number of cells. I believe it's a step forward, making cancer therapy more personalized by matching treatments to each patient's unique cancer profile.
Thanks so much, Maryam. That's a very good reasoning. And you sort of answered what my second question was going to be, but maybe what was it that brought you to pursue this area of research? Was it a disease interest, or did it just come around randomly? You met the right professor who you'd like to work with. How did you find yourself in working on this topic?
Well, I definitely met the right professor and right project at the right time. But I would say I was motivated by the gap that I see exists between what we know in research and how cancer is actually treated in clinic. So some patients still receive standard treatments that may not work for them personally. And that's the reason I became interested in functional precision oncology, because it aims to find the best treatment options for each patient before actually exposing the patients to those treatments. And the idea that this could one day help personalize treatments and reduce side effects really inspired me.
That's really great to hear. And maybe I know a little bit about Silvia's project, so and how actually important it is. But maybe Silvia, was there anything in your project content that inspired you to take up this research topic?
Yeah, actually, I was working in high throughput screening before I joined Fraunhofer. And then, yeah, for me, it's the same. I met the right people at the right time when I was motivated to change something in my life. And then there was this project with patient-derived models and 3D drug screening, which wanted to be set up. And then I stepped by, and this really motivated me because it was challenging and interesting. And also what Maryam said, for the patients to have the option to find the better treatment, like the standard treatment, maybe which doesn't help and so on. And this was also a big motivation for me.
Yeah, and I think I can certainly align on that. Some of us who've been doing this for a little bit longer know that sometimes a very simple assay, although it's easy to do and you get high quality data, actually you don't have that translational capacity in the assays, and you have to really invest time and effort to run complex assays with complex endpoints, but that really have the chance to bring those compounds forward. So it's really great to hear what you're doing there. So Toan, just the same kind of question to you. What really kind of inspired you to get involved in your work?
So in my master's projects, I work a lot on organoids, especially the cellular organoids. And as you know, for the organoids, the common extracellular matrix that we need to use is the materials. So, but material has the disadvantage such as it's expensive and it have batch-to-batch variation. So when I continue my PhD project, so the motivation that I want to find alternative hydrogel that can replace material. So in the current field is now the cellular extracellular matrix is emerging as an alternative for the material, because as you know, the material we extract from the sarcoma of the mouth, so it's from the tumor. It may not represent the cues of the native extracellular matrix. So we can develop materials from the cellular extracellular matrix can better mimic the microenvironment of their tissue. And from there, then it evolved into building an organ-on-chip system to model the disease like automucositis, which still lack effective treatment. And I was inspired by the possibility of combining biology and engineering to create something clinically meaningful.
Yeah, fantastic. That sounds. I think that matches many. I think the motivations of the people that I'm aware of in the field. So just moving on to maybe to a question to Lan about while we were doing your research topic, what was the kind of most surprising or unexpected observation that you made or finding that you made, something that you couldn't have predicted beforehand, or did everything run smoothly? How did it go for you, Lan?
I think that's really a great question. Because as I introduced before, that I am constructing some RNA-based biosensors, and I mean for my project, when I was like trying to develop this kind of RNA-based bioluminescent sensors, I think the most surprising thing that's unexpected to me was probably how significantly in the secondary structure of the RNA could affect its in vivo performance. Because we know like everything that worked in vitro will probably not gonna work in vivo, and there's a lot of like conditional optimization that you have to do to make it be transferred perfectly from in vitro experiments to intracellular or in vivo experiments. So I actually learned a lot from the sensor design, that how the in vitro conditions could affect the like behavior of the sensors, and how will I be able to like maximize the function of the in vitro developed sensors for any intracellular detections or like functions studies. So yeah, that's basically what I've learned, and something that's unexpected during my project.
Yeah, that's. I think that there's many unexpected discoveries that sort of later turned out to be drugs. I used to work at Pfizer, and we have a classical example of that with yes, well, one of our drugs, Viagra. But that happened during my time at Pfizer. So maybe Silvia, I've got a kind of question about the conference experience itself, and how do you think going to an SLAS meeting did that sort of shape or change your understanding? I know you've worked in the industry before, but how was the experience at the meeting in Hamburg, and how did that go for you?
I mean, the experience was really great to have all these different, interesting topics there. And especially for me, as I mentioned, I was working in the HDS business before, and basically more 2D assays and biochemical assays, and it was really high throughput. And what I experienced during the last years is that's also somehow going back to a lower medium throughput, because the assays become more and more complex. And this is what I also experienced, that the industry is doing also these downscaling but more complex assays. And also what I experienced then in the context with my poster is that it's really, of use. Also a great experience, that what I did is useful for others, not only for my PhD project.
Yeah, no, that's great to hear. And maybe Tuan, you were the international conference is still quite a lot larger than the European conference, although we're trying to catch up. But how was the experience for you being with all of the technology providers and the talks and all of the workshops and those sessions? But what did you sort of learn from that about how the world of automated drug discovery and high throughput screening and the application of automation in life science? What was the main thing that you took from that experience?
So SLAS was really a game changer for me. So it broadened my perspective beyond the dentistry field that I came from, and it helped me to see my work through the lens of translational science and also drug discoveries. So I received many feedback from researchers in pharma and tech, and which challenged me to think more critically about scalability, reproducibility, and application in a screening platform. And also being a finalist in the student poster competitions gave me the confidence that my work has value beyond academia.
No, that's excellent. I mean, we all, I think, in the medicines discovery world, it's absolutely dependent on basic research taking place in academia, but it's really industry that gets drugs to the market, and having an interest from industry in your work and them potentially picking it up and using it. I think that's always a great thing. But Maryam, you come from the KIT, as I understand, and that's one of the leading technology universities in Europe, I think. And how did all of the technologies that you were exposed to during your time at the SLAS meeting, did they impress you, or were you used to this kind of stuff coming from the KIT? How was your experience?
It was definitely very inspiring. I was inspired by how much innovation is happening in the field of automation, drug discovery, and also data analysis as well. And also presenting my work helped me realize that our approach here in KIT, how can it fit into a much larger trend of making drug discovery faster and more efficient. And so I would say that that made me more aware of what and where is the field going, and how, for example, my work can contribute to that future.
Oh, that's great to hear. So you weren't the first, and you won't be the last students looking to sort of bring their work in the form of posters to the SLAS. And maybe a question for Lan. If you had to give some advice to other students on creating an excellent poster, which is like the one you have, what advice would you give to students preparing a poster or getting ready to come to an SLAS meeting in order to get the most out of it?
That's a good question. I think my main advice would be, probably the most important thing would be the clarity and simplicity for like to go along for your poster presentation. And like as an international conference like as SLAS, I think they'll bring a very wide audience. Like some of the audience are experts that's in your area of research, and some are not. So I think our poster should tell a very clear story that anyone can follow in just a couple of minutes, and also try to avoid some overcrowding like text in the poster or your raw data. Try to use some visuals like schematics or graphs to guide people through your work. Also, probably if you have time, you can practice a pitch in front of your colleagues or your friends, probably just a 1 to 2 minutes summary of your project. So we would be ready to engage with people quickly and meaningfully. And I think last but not least, don't be afraid to initiate like this kind of conversations during the meeting, because often the most valuable interactions happen when we are like initially proactive and open to some feedback from the audience, and that's how the conversation actually starts, and how you would like start like introducing your poster.
I completely agree. The only way we learn in these types of settings is talking about the problems that we have, and seeing if those around us and the other attendees at the meeting, if they've had the same experiences, and then we can, adjust what we're doing and improve. I mean, the poster is a kind of picture of a research activity, but it's the conversation you have with the person walking past. That's the really important thing, and that's where I think we can learn the most. But Maryam, from your side, what would be your advice? Sometimes Europeans are a bit more reserved in their when they're in going around the posters. What was your experience to engage with the people at the poster session and show your work?
Well, I find it very interesting that actually at SLAS people come from different disciplines. So they come from, for example, automation, chemistry, biology, data sciences, all in one place to you. And then you can have a deep conversation about them. And also it helps to look at your work and your poster from different perspectives. And it sometimes even helps to, for example, see something in your work which is missing, or it can inspire you what kind of other applications your work can have. And also in my personal experience, I find it useful to practice explaining your work, both in technical and in simple terms, because as I just said, not all people who you will meet come from your disciplines, and it can be hard for them to maybe follow your work.
Yeah. Yeah, I think that's a good part. I try to make a point of looking at posters where I don't really know what's going on, so I can learn something as well. Even at my advanced age, it's important for me. The posters are a great opportunity for that. So maybe Silvia, just to thinking about the future and what's the next step on your research journey then with your
Actually,
PhD and all those things. Yeah, what's happening?
so I'm still in the PhD phase, so I have to finish the writing and so on, and I hope this will be happening soon. And my journey actually started yesterday. I joined a company, and it's really interesting because
Congratulations.
I can apply my experiences to them, and I can also learn from them because they are somehow also doing related stuff, but not in the 3D, but 2D plus assays and so on. And it's really interesting, and I'm really looking forward. So for me, it's now really the application.
Okay,
of my
maybe I'm gonna ask a nosy question, but did you meet that company at the SLAS
Yes.
meeting or did?
Yes.
Wow. Okay, this is not
But,
a stage. That's fantastic. Yeah, very good. Okay, it's then yeah, things are really happening on your side. Maryam, what's next in line for you then? After you say you've just completed your PhD, where are you heading now?
I also have to wrap up the project and write an interesting paper based on that. Also, I'm looking for new opportunities. And since I enjoyed working across disciplines like biology, automation, and data analysis, I hope I can find a project, whether in industry or academia, that can also help me to keep growing in this space. Also, I would like to look for an opportunity where I can learn and contribute to meaningful projects and be a part of a collaborative and dynamic research environment.
Yeah, sounds great. And Lan, for you, how does the future hold? Will it involve future SLAS meetings, do you think? And where are you heading now?
I think it's a tough question for me because I really wanted to attend a future SLAS conference, but since this is my last year of PhD study, not sure. And also I'm like currently actively looking for jobs, and I really wanted to and get a job in like the biotechnology sector, like get a job in a biotechnology company. And I wish that I can focus on some RNA-based therapeutics or imaging-driven drug screening. To be honest, I think attending the SLAS International Conference for the first time was really a eye-opening experience for me. And I was actually I was really struck by the level of lab automation on display, and everything from automated liquid handlers to fully robotic high throughput workflows. It kind of made me realize that how transformative automation is becoming in scientific research. And I feel like as a PhD student right now, I really rarely had a chance to interact or ever seen this kind of technology, to be honest. So seeing it firsthand broadened my perspective, and that kind of changed my now I'm not sure change my idea for finding a job in the future. I used to really wanted to go to academia, but right now I was maybe get a job in a company that's doing some automation, fancy stuff, or
Yeah.
kind of robotic stuff that's really really excellent. And I believe working in this kind of fields would probably not only be meaningful to my own professional career growth, but also I am wishing that I can contribute to the entire human society for a broader scale, some kind of stuff like that. But I am just thinking my future could end up something like that.
Yeah, I know that's a very good point. If we look at everybody hypes up AI and machine learning and all those things, these days as being enablers of the drug discovery or the next animation on Netflix. There's a whole areas of application, but they're all highly dependent on access to well-defined, high-quality data sets and data and the metadata that goes with them. And the only really sustainable and economic way to generate that data is through the use of automation. And I think that's this is the age of automation in drug discovery in particular. And it's not just about feeding algorithms, but it's really about, as you say, finding treatments for patients with unmet need. And that's certainly what motivates me. So last question to Tuan. Where's your scientific journey going to take you? You have a very nice picture of the Golden Gate Bridge behind you. What's on the other side of that bridge?
The other side of the bridge is my messy room. So for the future, I hope to continue developing the organ-on-chip system for disease modeling and personalized medicines. And I'm especially interested in integrating the patient-derived cells to explore their potential in predicting drug responses. I will graduate my PhD next year, so I plan to return to Vietnam and apply for a position at a university, so where I can build my own lab and continue to bridging biomaterials, tissue engineering, and microfluidics. And recently, I'm very interested in develop an human on chip, like combining many organs together on a chip. Yeah.
That sounds great. And thank you for all of your kind of sort of inspiring ideas of the future. Maybe a small anecdote from my side. I remember 25 years ago when I started in screening, and one of the computational guys told me, "Oh, you guys are going to be out of business in a few years' time because we can predict everything." And 25 years later, we still have to do experiments. We still have to reflect the complexity of human disease and human biology in a meaningful way, whether it's in a microtiter plate or on a cover slip or in an animal model. So I think there's opportunities and roles for all of us to contribute to that. And it's been really amazing to hear what your journey's been, and also, hopefully the way that the SLAS and engaging with the SLAS has sort of helped you along that journey. And the SLAS is there for you in the future as well. We're a member society. We're here for the benefits of a member, for their education and their professional career development. And I hope you continue to engage with us and come to future conferences. And I look forward to maybe listening to a podium presentation or an invited plenary in 10 years' time when all your research objectives come to fruition. So thanks so much for your time today. And we'll hopefully see you soon. Thanks a lot.